Okay, hello everyone. Let's continue with the story on carbohydrate catabolism. So, if you remember, we have talked about digestion and absorption of carbohydrates, and we talked about the first catabolic pathway of glucose, which is glycolysis, and now we have to continue further on with the degradation of glucose molecules with the next metabolic pathway, which is oxidative decarboxylation of pyruvate. So let's start. Uh, if you remember our last video when we talked about glycolysis, the final product of aerobic glycolysis is pyruvate. So now we have to see what is going on with it. So what is the fate of pyruvate? If you take a look at this figure, you can see that there are a lot of possible pathways. So 6 different pathways, different fates. Which one should one molecule of pyruvate choose actually depends on the current metabolic needs of each cell. So let's start and list all of these possible 6 pathways. So this is the figure representing the possible pyruvate fates, and let's start and list and explain all of 6. So let's start with the first one. So this is something that we know very well. The last step of aerobic glycolysis is conversion of phosphoenol pyruvate to pyruvate. But what about the reversible process? What about the conversion of pyruvate to phosphoenol pyruvate? We can say, and we mentioned earlier, that this is not a direct or indirect or reversible reaction. So it is not possible to do it reversibly using pyruvate kinase as it was in glycolysis. So in order to convert pyruvate to phosphoenol pyruvate, we have to do one additional step by synthesizing oxal acetate and then decarboxylating oxal acetate to phosphoenol pyruvate. So it occurs via oxal acetate, and we're using ATP for those purposes. So having this in mind, it is an open possibility to synthesize glucose molecules from some other metabolites, which are non-carbohydrate metabolites, from fatty acids, from acetyl coenzyme A, from amino acids, and so on. But more detailed insight in the events which takes place in such a process, we're going to talk about it in the gluconeogenesis pathway. So this is one of the possible fates. So conversion of pyruvate to phosphoenol pyruvate via oxal acetate in the metabolic pathway called gluconeogenesis. Okay, so let's move on. Which one is the next? This is also something that we know very well. So conversion of pyruvate to L-lactate in anaerobic conditions in human cells by the enzyme lactate dehydrogenase, the reaction in which one NADH is oxidized to NAD+, and this is the reaction which takes place and occurs during intensive muscle work or muscle exercise. And we have discussed about this reaction previously. Let's move on. This is one very interesting reaction. Conversion of pyruvate to ethanol to alcohol, which occurs in anaerobic conditions. It can occur in human cells, and also it occurs normally in microorganisms. So it is the reaction of decarboxylation, sorry for the typing mistake. So it's the reaction of decarboxylation of pyruvate by pyruvate decarboxylase to get an acetaldehyde. Acetaldehyde can be then reduced by alcohol dehydrogenase to ethanol. So this reaction when occurring and taking place in microorganism is called fermentation or alcoholic fermentation, widely used during the production of wine and beer. But in human organism, this is why I'm mentioning it, it's the phenomenon which is found in some people, in some persons, which are capable of converting pyruvate to ethanol. So certain amounts of ethanol can be found in those persons' blood despite the fact that they haven't taken any alcoholic beverages at all. And this syndrome is marked as self-brewery syndrome. So let's move on. What is the next option for our pyruvate? The pathway number 4 is conversion of pyruvate to L-alanine. Where pyruvate is a kind of keto acid to L-alanine. So in the reaction, in the pathway called transamination, pyruvate can accept amino group from some other amino acids to become an amino acid itself, alanine, and amino acid is converted to keto acid. So this is the process of transamination. In this case, conversion of pyruvate to alanine and opposite, the reaction is reversible, is catalyzed by alanine aminotransferase, ALT enzyme. I hope that you have heard about this enzyme so far. And this is something that you're going to talk in more details during the story on catabolism of amino acids. For now, just to list that this is one option for pyruvate conversion in our organism. Let's move on. This is the 5th pathway. So conversion of pyruvate to oxaloacetate. And this is the reaction of carboxylation. So one carbon dioxide or bicarbonate ion is incorporated in the molecule of pyruvate by the enzyme pyruvate carboxylase. This is also the reversible reaction and requires biotin for its function and ATP for the synthesis of new chemical bond. Oxaloacetate, which is formed by this reaction, can further be transferred to citric acid cycle, to Krebs cycle in order to obtain certain amount of energy, or in the process of transamination it can be converted to aspartate in the reaction catalyzed by AST, aspartate aminotransferase. And finally, this is what is gonna be our task today, the 6th pathway, the last but not the least, conversion of pyruvate to acetyl coenzyme A, which is the reaction of decarboxylation, which means the removal of one molecule of carbon dioxide. This reaction is irreversible, and acetyl coenzyme A obtained in this reaction, in this metabolic pathway, may go to Krebs cycle to produce ATP and carbon dioxide and energy, or it can be redirected to fatty acid synthesis, again depending on the current needs of a cell. But this is going to be the pathway on which we are going to focus our attention at this very moment. So oxidative decarboxylation of pyruvate is a pathway which occurs totally in mitochondrial matrix. So you know that pyruvate is formed by glycolysis. Glycolysis occurs in cytosol. So pyruvate first has to pass from cytosol to mitochondrial matrix. This is achieved by protein carriers incorporated in the inner mitochondrial membrane, and the mechanism of the transport of pyruvate from cytosol to matrix of mitochondria is co-transport, usually with a symport with some ions, for example hydrogen ions or something similar. The reaction is catalyzed by one enzyme complex which is called pyruvate dehydrogenase, and this is the overall reaction of the pathway. So pyruvate is converted to acetyl coenzyme A. So we have the usage of one coenzyme A, removal of one carbon dioxide, and since this is a redox reaction, we have the removal of reducing equivalents and formation of reduced NADH coenzyme. So if you take a look at this reaction, it looks kind of rather simple. However, because there are millions, however, in biochemistry, this reaction is not that simple. On contrary, this is an enzyme complex of pyruvate dehydrogenase, which is a complex system containing 3 enzymes and 5 coenzymes. So before we move on to the steps of this pathway, I would like to list those 3 enzymes and 5 coenzymes first, which means first I like to say which ingredients we need for the pathway, and then we're gonna start cooking. So enzymes which are involved in this pathway are pyruvate dehydrogenase, or I prefer the name pyruvate decarboxylase in order just not to mix up the name of this first enzyme with the name of the overall of the total complex. So let's name it as E1. The second enzyme is dehydrolipoyl transacetylase, or E2, and the third enzyme is dehydrolipoyl dehydrogenase, or E3. Let's list those 5 coenzymes. First, thiamine pyrophosphate, TPP, then lipoic acid, LP, coenzyme A, CoA, FAD, flavinadenine dinucleotide, and NAD+, or nicotinamide adenine dinucleotide. Before we move on with the pathway and the reactions, let's just remind ourselves in brief about these coenzymes and their structure. So TPP, thiamine pyrophosphate, originates or is synthesized from vitamin B1, containing aminopyridinium ring and thiazolium ring, and this is the carbon atom of interest. This is an acidic carbon atom, which is reactive center of this TPP, and this carbon atom and its acidic hydrogen attached to it is actually capable to bind to itself certain atomic groups, which we are going to see later on. A lipoic acid also has a unique structure. It is involved in redox reaction because it is capable of accepting or donating reducing equivalents, because there is one specific part of its structure. This is disulfide part of the structure in oxidized form of lipoic acid, and sulfhydryl, 2 sulfhydryl groups in reduced form of so-called dihydrolipoic acid. Then we have coenzyme A with its rather complex structure, as you can see, including ADP and pantothenic acid, and of course the capability to form thioester bonds, which are high energy bond, and this is the structure of acetyl coenzyme A, which is one of the most famous forms of coenzyme, activated coenzyme A. Then flavinadenine dinucleotide, which is also a coenzyme of oxidoreductases, consisting of flavin or isoalloxazine ring, with ribitol sugar alcohol attached to it, an ADP molecule. So on isoalloxazine ring, reduction or transfer of 2 electrons and 2 hydrogens occur. So FAD is converted to FADH2, its reduced form. And finally, NAD, nicotinamide adenine dinucleotide, with the amide of nicotinic acid as one nitrogen base and adenine as another nitrogen base, so it's dinucleotide, so 2 nucleotides attached by pyrophosphate bond. So this is an oxidized form where nitrogen is plus charged here, and it can accept reducing equivalents, 1 hydrogen to heterocyclic ring and one proton apart. So NAD plus oxidized form, when reduced, goes to NADH plus H plus. So now when we have remind ourselves about the structure of coenzymes, let's see the pathway. So this is the overall pathway of oxidative decarboxylation, and let's do it step by step with a total of 5 steps. So let's start with the step number 1. So if you can see from from the figure here. So pyruvate actually undergoes decarboxylation. So one carbon dioxide molecule is released from the system, remember it. And the rest of the molecules, and that is hydroxyethyl derivative. So hydroxy and two carbons, ethyl, hydroxyethyl derivative is formed, and it's attached to TPP, to reactive carbon atom on TPP, which we've previously mentioned. And the enzyme which catalyzes this reaction is pyruvate decarboxylase or pyruvate dehydrogenase. Let's move on to the second step. So this hydroxyethyl intermediate is further oxidized. So this hydroxyl group is oxidized to keto group. So hydroxyethyl derivative is converted to acetyl derivative. And during this process, this acetyl derivative is transferred from TPP to lipoic acid, disulfide form of lipoic acid. And this transfer is catalyzed by dihydrolipoyl transacetylase, which means transfer of acetyl group to disulfide form of lipoic acid or oxidized form of lipoic acid. So what we have here? Here we have an acetyl group attached by thioester bond to SH group of reduced form of lipoic acid. So this is the high energy bond, so acetyl group can be transferred to coenzyme A in the step number 3 to form acetyl coenzyme A, which is one of our final products, and this time reduced lipoic acid is released from the system. So far we have carbon dioxide form, we have acetyl coenzyme form. So now we have reduced form of lipoic acid, but now this is dihydrolipoyl, so we have to regenerate lipoic acid, which occurs in the step number 4. So sulfhydryl form of lipoic acid is now oxidized back to disulfide form by FAD-dependent dihydrolipoyl dehydrogenase, which is the third enzyme, which ends up in the regeneration of disulfide form of lipoic acid, and these reducing equivalents are transferred to FAD coenzyme, which becomes reduced FADH2 form. And we can do the similar thing again. We have to regenerate FADH coenzyme, which is gonna occur in the reaction step number 5. So FADH is reoxidized to FAD again by dihydrolipoyl dehydrogenase, which yields the formation of reduced NADH coenzyme, which quickly enters electron transport system, which yields the formation of ATP, if you remember. So these are 5 steps of this metabolic pathway, yielding the final products, as we've seen, acetyl coenzyme A as the major final product, carbon dioxide in the step number 1, and ATP, which is derived from NADH coenzyme, and we know from our previous general chemistry course that one NADH coenzyme in electron transport system forms 3 molecules of ATP. So if we want to calculate the energy yield, so if we have the formation of one NADH in one rotation of oxidative decarboxylation, which means 3 ATP, so we can conclude that the energy yield is 3 ATPs per molecule of pyruvate. But if we take into consideration that we are degrading glucose molecules and that we obtained 2 molecules of pyruvate at the end of glycolysis, which means 2 pyruvates enter oxidative decarboxylation, which yields the formation of 2 acetyl coenzyme A's and 2 NADHs, gives us the total of 6 ATPs per molecule of glucose. What happens with acetyl coenzyme A upon its synthesis? a major portion of acetyl coenzyme A actually enters Krebs cycle. Acetyl coenzyme A can also be used for fatty acid synthesis and 3-acylglycerol synthesis if the Krebs cycle is saturated, or if a cell is saturated by energy by ATP, or it can be redirected to amino acid metabolism. Which of these three pathways is going to happen? That depends, I'm repeating again, on the current metabolic needs of a cell. So how this pathway is regulated? So there are two ways to regulate oxidative decarboxylation of pyruvate and this complex containing 3 enzymes and 5 coenzymes. So first, it's inhibition by reaction products or feedback inhibition, and 2, covalent modifications of enzymes by phosphorylation and dephosphorylation, which is one of the common pathways to activate or deactivate enzymes, if you remember it from our general biochemistry course. So in this figure, you can see how this is achieved in the case of pyruvate dehydrogenase complex. So let's analyze this figure. Pyruvate dehydrogenase complex is active when it's dephosphorylated. Upon phosphorylation of this complex, inhibition occurs and it becomes inactive. So phosphorylation of this complex is achieved by pyruvate dehydrogenase kinase, and dephosphorylation is completed by pyruvate dehydrogenase phosphatase enzymes. These two enzymes are affected by a number of components. For example, ATP as a marker of sufficient amount of energy in a cell, or accumulation of acetyl coenzyme A, or NADH coenzymes, which are all products of oxidative decarboxylation of pyruvates. Here they are going to stimulate and activate the activity of PDH kinase, which is going to stimulate the phosphorylation of pyruvate dehydrogenase and slows down the process. So if you have a lot of energy in a cell, a lot of acetyl coenzyme A or reducing equivalents, this is going to be a signal for this pathway. Okay, let's phosphorylate, let's slow down a little bit. On the other side, if there is an increased influx of pyruvate into the system, of course, if you're introducing one of the reactants, is going to move or shift the equilibrium towards the formation of products, which means that pyruvate is going to inhibit PDH kinase, so pyruvate dehydrogenase will remain active, thus speeding up removal of pyruvate and its conversion to acetyl coenzyme A. On the other side, calcium ions can promote the activity of PDH phosphatase, which is going to activate this PDH complex and the degradation of pyruvate. Of course, NADH and acetyl coenzyme A can directly inhibit PDH activity, not only via PDH kinase activity. This is summarized in the table provided in this slide. So activation of the complex is achieved by ADP, NAD+, pyruvate, and calcium. And these are the mechanisms of the effect expression: decreases kinase activity or decreases kinase activity and increases phosphatase activity. Insulin increases phosphatase activity. Inhibition of the complex is achieved by ATP, NADH, and acetyl coenzyme A, which all increases kinase activity, and the last two, they inhibit the action of these enzymes by the product or by the mechanism of feedback inhibition. So more or less that would be all regarding this pathway, oxidative decarboxylation of pyruvate. So the next pathway which is going to be on the menu is one of the common fates of acetyl coenzyme A, and that is its conversion, final conversion to carbon dioxide and water and production of ATP, which is going to be the famous maybe the most famous pathway of all, which is Krebs cycle. But until that time, but until that moment, this is time to make a short break, and until next time, bye bye. Thank you.